Hello everyone, and welcome to today's Lab Tools webinar with The Scientist. I'm Betsy Young, Director of Creative Services for The Scientist, and I'll be moderating our discussion. Today, our speakers Scott Schachtel and Kevin Flynn will be discussing how iPSC-based 3D tissue modeling has become a popular in vitro option for disease modeling in spite of some challenges with variability. As representatives of our sponsor, Bio-Techne, they'll also introduce their solution to this problem, the MimEX Tissue Modeling System. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Ask a Question tab and type your query into the question box located on the bottom left of your screen. We'll try to address as many of these questions as we can during our Q&A session. Our webinar platform is user-friendly. You can expand the presentation window by simply clicking on the diametrically opposed arrows in the upper right-hand corner. This will maximize the display within your screen. The webinar will be archived on the Scientist website, and we'll send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor. Bio-Techne combines the best-in-class products and services from R&D Systems, Novus Biologicals, Tocris Biosciences, Advanced Cell Diagnostics, and Protein Simple, making Bio-Techne and its brands the best strategic partners for neuroscience researchers. Bio-Techne has a vast product range and manufacture over 90% of their bioactive proteins, application-qualified antibodies, ELISAs, LumenX assays, small molecule inhibitors and activators, instrumentation, and RNAscope technology. Our sponsor has provided us with some helpful resources on in vitro 3D disease modeling, and you can download them at any time during today's webinar. You'll find them on the left side of your screen. And with that, let me introduce our first speaker, Scott Schachtel. Dr. Schachtel is the product manager for the stem cell and cell culture portfolio at R&D Systems, a Bio-Techne brand. Scott received his PhD in neuroscience at the University of Iowa, where he used multiple primary cell culture models to investigate neuronal and auditory synapse development. During a postdoc in neuroimmunology and neurovirology at the University of Minnesota, he investigated the effects of cytomegalovirus infection on neural stem cells. He's been at Bio-Techne for 4 years, where he works closely with their scientific teams to bring unique and innovative cell culture solutions to the research community. Dr. Schachtel?
Yeah, thank you very much, Betsy. And I'd like to take this opportunity to thank the Scientist team for allowing us to present this great material to the audience, and also to the audience, people who registered and who are in attendance now, for joining me today. So the title of my talk today, or our talk today, is really evaluating new 3D systems for disease modeling, toxicology, and drug screening. And the way we're going to do that is through just walking through some of the cell culture models that are being used currently for toxicology and disease modeling. And we're going to divide it between Kevin and I. So I'm going to start by providing an introduction about models that are currently being used and why they're being used, and some maybe improvements in the field that need to be thought about when deciding on a model. And then I'll walk through cell lines and primary cell models and their benefits and challenges. And then I'll talk extensively about the use of stem cell-derived models and some of the considerations and tools that Bio-Techne has that can help improve efficiency for your research. And then I'll hand it off to Kevin, and he'll expand in more detail on 3D and organoid cell culture model systems and present to you an innovative model that we've developed in-house called the MimEX Tissue Model Systems. So before getting into the meat of the talk, I just wanted to give a brief overview of Bio-Techne. I know that Betsy just gave a little bit of a brief one, but really I wanted to present to you why Bio-Techne provides great solutions for the cell culture workflow. So really bringing together these life science brands of R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, and Advanced Cell Diagnostics, we can provide many tools across the cell culture workflow for you, from proteins and growth factors and small molecules and media supplements to culture your cells, to antibodies, assays, and protein instrumentation to analyze your assays and your experiments. Recently, we acquired a couple new companies, the first being Trevigen, which brings to our portfolio a wide variety of DNA damage and cell damage assays, as well as extracellular matrices and basement membrane extracts that are commonly used for 2D and 3D cell culture systems. We also recently acquired Atlanta Biologicals, which brings to our portfolio fetal bovine serum, other sera, and cell culture media and reagents. So on this slide, I just wanted to illustrate kind of how all these brands come together to provide you with a workflow solution to your cell culture. So this is just a general schematic of 2D and 3D cell culture workflow, breaking the steps up into 4 kind of basic steps from isolate and culture, verification, differentiation, and investigation. And just to give you a glance at the 10,000-foot view of what kinds of categories of tools we have for you, again, from proteins to assays. And with that brief introduction of the company, I'll move on to kind of the meat of the talk here, starting with an introduction on pharma research, development, and manufacturing workflow, which is illustrated on this infographic here. And really the point to illustrate here is the complexity and the length of time that it takes to bring a drug through research and development all the way through clinical trials. And it's really not only just the complexity and the time, but there are a lot of resources put into the different phases, as well as, importantly, money. And really the cost to develop compounds has grown astronomically in the last decade or so, and this is alongside the actual increase in failure rates of drugs that are being brought through the development pipeline. So the histogram on the right is taken from a review article in Nature that was looking to assess the reasons why clinical trials were getting suspended during the late stages of the clinical trial. And the takeaway from here is that the majority of the failures were attributed to failure in efficacy and safety. And so that sort of harkens back to maybe reassessing at the research and development phase what kinds of model systems we're using to both assess efficacy and toxicity. And so if we look at the research and development workflow for drug development, toxicology is a critical component of this. And so where does it fall in that timeline? So after you've identified a target, have a hit, you're going quickly into in vitro model systems to assess the potency and cytotoxicity of that compound. So doing a range of things, looking at pharmacodynamic properties, as well as doing ADME tox property assessment. And those will be done before you move into animal studies or going into clinical trials. So really what's important to optimizing success at the clinical phase, this is really dependent on identifying appropriate and robust in vitro model systems during the early phase of research and development, and then choosing quantitative data endpoints during that research and development phase that can be used and that are predictive for use during the clinical phase. But for the purposes of the talk today, I'll focus on identifying appropriate and robust in vitro models. So this slide illustrates on the bottom cartoon, stretch of cartoons there, the types of model systems in life sciences that are currently being used to model disease and disease modeling and toxicology modeling. And this is juxtaposed to kind of the complexity of the human body on the top. So with more simple systems on the left, like monolayer cell cultures, to more complex systems on the right, such as multiplexed organ-on-a-chip models. And we can kind of generalize the spectrum here of the model systems for the models on the left being more simple, with the benefits of being more accessible and more homogeneous and standardized, which are really important factors for decreasing variability when you're starting to work in high throughput systems. However, the predictability of these models has come into question as of late. And then as you move over to the right side of the model system spectrum here, the models are getting more complex. They're looking more like in vivo-like systems, and therefore are thought to be more predictive in their assessment of how drugs may interact with the tissue. However, a trade-off with this is that these systems tend to be more difficult to culture and to analyze. So really, as you're considering the model systems that you're going to work with in your disease modeling or toxicology screening, you have to consider both the complexity of the model, but also the cost-benefit of the analysis and setting up the culture system. So the next sections of my talk, I'll walk through some of the currently used models for toxicology and disease modeling, starting with cell lines and primary cells, and then talking more extensively on stem cell-derived models. So cell lines are defined as cells that can be continually passaged and that display genotypic and phenotypic homogeneity. And the graphic on the right here illustrates some commonly used cell lines for each of the organs listed, and the infographic there with the ones in red being the most common ones that are used in the literature. And so the benefits of using cell lines in general is their ease of culture and rapid expansion ability in culture. They also display a homogeneous phenotype, which is good in that as you set up your high throughput experiments, this will help reduce variation in your inter and intra experimental variation. And then they're also customizable, so you can transfect these cell lines with a protein of interest and look at drug-protein interactions specifically. However, some of the benefits of cell lines are also their challenges. The homogeneity of cell lines is farther removed from the in vivo microenvironment, so the accuracy to native biological responses that they give will be maybe less predictive. Also, there's inherent genetic drift in cell lines and potential for cell line contamination. An important factor is that these cell lines are typically cultured in a 2D monolayer, which again is not akin to the microenvironment that the drug might be interacting with in a human or in a tissue. So primary cells are a little step closer to a more in vivo-like situation. Primary cells are cultures started from cells, tissues, or organs taken directly from an organism of interest. The benefits of this system is that because they're taken from an organism of interest or a tissue, these cells are physiologically more normal and retain phenotypic and genotypic characteristics of the cells found in vivo, making them more biologically relevant compared to cell lines. Primary cells can also be passaged in culture, so you can establish a system like that. Oops. Some of the challenges with working with primary cells is that really the access to the tissue and the isolation steps. So gaining access to the tissue can be difficult, as well as during isolation, cell yields can vary depending on the tissue size and the isolation protocols that you're using. Now these cells can be passaged in culture, but this is finite, so they have expansion limitations, and this is due to the fact that the cells will senesce over multiple passages, and then a new source of primary tissue will be needed. And again, just like cell lines, these primary cells are typically cultured in 2D monolayers, which again is farther removed from the 3D microenvironment that a drug may be interacting with in vivo. Okay, like I said, that was going to be brief, and I'm going to spend the rest of the talk speaking about stem cell-derived models, which have become rapidly adopted in the fields of disease modeling and toxicology over the last decade or so. And so when I speak of stem cell-derived models, what I'm really referring to are models using induced pluripotent stem cells as a cell source. And so in the image here, we see a green induced pluripotent stem cell colony grown on mouse embryonic fibroblasts. But really the benefits of stem cells are their characteristics that define them. So their ability to proliferate in an unlimited manner, and their ability to differentiate into all cell types of the body. And so what really combining these two facts together makes them able to provide an unlimited source of terminally differentiated human cells for use in disease modeling and toxicology. So kind of combining the benefits of cell lines and primary cells. Another benefit is the ability to derive patient-derived induced pluripotent stem cell lines, giving the capability of doing personalized medicine using these cells. Some of the just outright challenges with working with pluripotent stem cells, again, typically they're historically cultured in 2D monolayer types of systems, and starting with induced pluripotent stem cells, the terminally differentiated cell types tend to have a more immature phenotype than what you would find in adult cells from the tissue. But a recent challenge, or not recent, but a challenge that's come more to the forefront in recent years is trying to accommodate the cell line variability between induced pluripotent cell lines, and as well as trying to come to and develop standardized protocols for differentiating these cell lines into different cell types. And I'll talk more about those in the next few slides. But first, let's take a look at the stem cell workflow for high throughput toxicity screening. So demonstrated here on the top is isolation of fibroblasts or peripheral blood mononuclear cells from a healthy or diseased individual. These cells are then reprogrammed into induced pluripotent stem cells using a number of different methods that are out there, including transcription factor overexpression or small molecule treatment. Once you've verified and validated your pluripotent stem cells, you then expand them and differentiate them into the cell type of interest on the bottom. These can then be translated into high throughput screening models for toxicity and drug screening. And in the case of personalized medicine, the potential to select a particular treatment that's specific for the individual from which the original cells were isolated from. But if we talk more generally about using stem cells for high throughput screening, we're looking at more like the bottom half of this workflow, where you're establishing pluripotent stem cell lines on the right-hand side and using them as an unlimited source to expand and create different cell types of interest for testing, which then can be applied into high throughput screening systems. Some considerations to think about when using stem cell models for toxicity testing and disease modeling are making sure that your starting iPSC populations are optimized. And this can really affect the downstream differentiation efficiency and consistency. So making sure that that is optimized as well. And then when you have your terminally differentiated cell types, making sure that they express the right markers and are functional in the ways that we would expect them to be. And really what these 3 points kind of all point back to is really improving the reproducibility and efficiency of the stem cell system that you're using. The research scientists at Biotechne are intricately involved in developing and working with induced pluripotent stem cells, and they've developed a number of differentiation kits that can be used for toxicology and disease modeling. And these kits are StemXVivo Differentiation Kits, provide optimized cocktails of proteins and or small molecules that help differentiate pluripotent stem cells into terminally differentiated cell types such as cardiomyocytes or hepatocytes, or intermediary cell types such as neural progenitors. And in working with these stem cells in the lab here and developing these kits, as well as other things, the research scientists in the stem cell team have realized that really not all iPSCs are created equal. In this experiment, the team took 8 different iPSC lines that were created in-house and were assessed for their ability to differentiate into the 3 germ layers, ectoderm, mesoderm, and endoderm, as well as a couple different terminally differentiated cell types. And we can see For example, the KF cell line that was created was able to differentiate well into ectoderm and endoderm, but not so well into mesoderm and the other cell types tested. And this is compared to the JOY induced pluripotent stem cell line on the top here that was able to differentiate into the 3 germ layers quite efficiently, as well as all the other cell types we tested. And so you can see from just the extent of the grid that really there is a lot of variability in between different induced pluripotent stem cell lines, but also there can be variability in between different clones in one stem cell line. And so the team here tries to troubleshoot this and optimize our iPS cell experiments by doing a number of methods to validate our iPSCs prior to differentiation. Specifically, we do and assess marker expression using antibodies-based methods, but even more importantly, doing functional identification of our pluripotent stem cells to make sure that they indeed are pluripotent. And I'll just take a couple minutes here to walk through some of the examples of the verification steps that we go through. So the most basic one is really immunohistochemistry for pluripotent stem cell markers. This just shows a panel of pluripotent stem cell markers that we use to validate our stem cells, and we've released this as a commercial antibody panel that provides all these stem cell markers in one easy kit. One of our more popular tools to use to validate pluripotent stem cells is our human pluripotent stem cell live cell imaging kit. And what that consists of are fluoroconjugated antibodies for pluripotent stem cell markers. And these can be added directly into the cell culture media and incubated for 30 minutes. And then you rinse the media off, provide fresh media, and then you can observe the fluorescence directly under the microscope. Some examples of some of the images that we get here are displayed on the right. But what this really allows you to do is to have a quick assessment of pluripotency, the pluripotent makeup of your stem cells, without having to sacrifice a well to do so, and without affecting the ability of the stem cells to continue to expand or to continue to differentiate into downstream experiments. So on top of the marker expression analysis, we also do functional identification, so assessing pluripotency of the stem cells. The historical methods that have been used to do so are the embryonic body assay and the teratoma assay. And these are very lengthy protocols, and importantly, they're quite heterogeneous in the readouts that you get between experiments. And so to try to improve upon the pluripotent assessment, our team developed a human pluripotent stem cell functional identification kit, which includes growth factor cocktails that push the induced pluripotent stem cells into the 3 different germ layers, ectoderm, mesoderm, and endoderm, in just 5 days. And this slide shows some data from 2 different induced pluripotent stem cell lines that we developed in-house that were assessed for pluripotency using this kit. And you can see different staining for ectoderm, mesoderm, and endoderm here. But I think a really important point about this kit is that it helps standardize the pluripotent assessment of your stem cells. So you can compare across stem cell lines the ability of them to differentiate into the different germ layers. So for example, on the right-hand side here, there's a histogram where we did the functional assessment on 2 induced pluripotent stem cell lines, and we see that the Fab2 cell line has better differentiation into mesoderm. So if you were to want to do a differentiation down the mesoderm lineage, selecting the Fab2 cell line as your starting pluripotent stem cell population would improve efficiency of the system down the line compared to the ADLF1 cell line. Another observation that the stem cell team has made while they work with the induced pluripotent stem cells is that really the culture media can affect the ability of the iPSCs to differentiate. So this experiment, induced pluripotent stem cell line, was differentiated in 3 different commercially available media. and then assessed using the functional identification kit for pluripotency. And on the right here, it just shows that we found differences in the cells expanded in the different media, and then their downstream ability to differentiate. So to summarize the last few slides here, really optimizing your iPSCs for your experimental model starts with making sure that your iPSC population is optimized and efficient. So this can really dictate the downstream efficiency of and consistency of your differentiations, and can have substantial impact costs on the model, on your model, and really help control variability. Another important aspect to validate your cells is after you've done the differentiation, making sure that your terminally differentiated cell types are indeed expressing the markers they should and functioning the way they should. And to illustrate the lengths that we go through here at Bio-Techne when we're working with our stem cells, I'll use our StemXVivo Cardiomyocyte Differentiation Kit to walk through some of the protocols, or not protocols, but the things that we do to validate the cells after they've differentiated. So the StemXVivo Cardiomyocyte Differentiation Kit takes iPSCs and differentiates them into cardiomyocytes using a series of cocktails that are added along the sequence. And once we've got those differentiated cells, we typically validate them for marker expression, functional attributes. So in the case of cardiomyocytes, beat rate is the obvious one. And then looking at functional regulation. So will these cells respond to perturbations in the way that we would expect them to? And I'll just walk through a couple examples of each of these. So for marker expression, after we've differentiated iPS-derived cardiomyocytes, we're validating them for expression of cardiomyocyte markers such as troponin, but also looking at times at cardiomyocyte subspecific markers such as atrial and ventricular markers. We also look at physiological attributes. Like I said before, beat rate's a pretty obvious one, and we have some great videos on our website of beating cardiomyocytes produced with the cardiomyocyte differentiation kit. But with this static image, just trying to portray to you that we can assess cardiomyocyte beat rate in a number of different ways. In this example, using a calcium indicator dye to visualize contraction, and then using it to quantify and measure the contraction rate and the beat rate of these cardiomyocytes. In addition to beat rate, also looking at whether these cardiomyocytes can respond to known cardiotoxic insult. So in this instance here, using endothelin-1 exposure as a model for cardiac hypertrophy, and showing that iPS-derived cardiomyocytes here on the right graph, when exposed to endothelin-1, increase their expression of human AMP as expected. And then last, really looking to see if these cardiomyocytes will respond appropriately to known perturbations or cardiotoxic agents. So in this graph here, we're looking at unstimulated, in brown is the beat rate of unstimulated cardiomyocytes. So this normal cardiomyocyte beat rate patterns. And then we can add agonists and antagonists into the system to increase or slow down the beat rate pattern of these cardiomyocytes. So validating that they are responding like they should to agents that we're exposing them to. So really, I guess what I'm trying to hammer home with you guys is that using stem cell-derived models for toxicology, if you're doing so, really you can optimize the efficiency of these models by really looking carefully at your iPSCs that you're starting with and making sure that they have are optimized for the ability to differentiate into the cell types of interest. And after you've done that, then determining efficient protocols for the differentiation and making sure that those are standardized so you can limit your variability between experiments. And then really going through lengths to validate and characterize your terminally differentiated cells to make sure that they're expressing markers that they should and functioning and behaving the way that they should. And to conclude here, I just wanted to just kind of reiterate that there are a variety of models out there, 2D models such as cell lines, primary cells, and some stem cell-derived models that are currently being used for toxicology and disease modeling, and that the choice for this, of which model you're choosing, really is based on the application need, and sort of a balance between simplicity versus complexity and cost versus low cost versus high cost. Using stem cell-based models, really, if you're just doing it that, you should make sure that you're starting with optimized iPSCs and differentiation protocols. And if you're looking for more predictive models, you should consider looking at the more newer development of 3D model systems such as organoids and spheroids, which are becoming even more popular as more predictive models for toxicology. And with that, I think it's a great spot to hand off the reins to Kevin Flynn to discuss more in depth about 3D models and some of the tools that Bio-Techne has in that space. Thank you.
Thank you, Dr. Schachtel. As a reminder to our audience, you may ask questions at any time using the question box. Our presenters will have an opportunity to respond after our next speaker. And that next speaker is Kevin Flynn. Dr. Flynn is a scientist in the stem cell laboratory at R&D Systems in Minneapolis, which is a part of Bio-Techne. He earned his BS degree from Loyola University, Chicago in 2004, and his PhD from Colorado State University in 2008. He then moved on to postdoctoral work in Germany. During his postdoc, he received the Marie Curie Postdoctoral Fellowship. While his training was in the role of cytoskeleton in neuronal development and neural regeneration, Kevin also has experience in integrin biology, cell adhesion and migration in fibroblasts, keratinocytes, and stem cells. Kevin has been a scientist at Bio-Techne for nearly 2 years, where he's worked on optimizing cultures for naive induced pluripotent stem cells, as well as enhancing neuronal differentiations. More recently, he's worked on the isolation, culture, and differentiation of adult stem cells from epithelial tissues. Currently, Kevin continues optimizing adult stem cell cultures and differentiations, but also collaborates on projects dealing with pluripotent stem cells and neuronal cells. Dr. Flynn?
All right, so thanks for the introduction, Betsy, and I'm going to reiterate our thanks to the audience for taking time out of your busy day to spend some time with us on this webinar. So Scott gave a nice overview of using 2D culture systems to study cell and tissue physiology and provided solutions using stem cells and 2D cultures, which can yield great data with high throughput capabilities, but have the drawbacks of not fully reflecting the physiological environment. So I'm going to talk a little bit about ways to get at more physiologically relevant models, and this includes 3D and organoid cell culture models, which I'll cover in the first part of the talk, and then I'll talk about a system we've developed called MimEX, which is an alternative to these model systems. So 3D cell culture has been around for quite some time, arguably since the early 1900s with classic experiments by Harrison with spinal cord explants. But it's really in the 1970s where spheroids were developed using Chinese hamster lung cells, where the 3D cell culture models started to arise. And in the 80s, the techniques were further established by Cell and improved upon in the last 20 years. In the last 10 years, with the advent of organoids, there's really been an explosion in these 3D model systems. And the great thing about these 3D model systems is that they're much better at predicting drug efficacy and toxicology than 2D systems, as Scott was already talking about. And they bridge the gap between 2D cell culture and native tissues. So you can use these systems as an alternative to animal studies in many cases. And also, when you use human-derived 3D systems, they tend to be even more predictive than animal studies. Okay, so the benefits of 3D systems over 2D systems are pretty obvious based on the organization of the cells in the tissue. So in 3D cell culture models, they are interacting with other cells in 3 dimensions. So in X, Y, and Z, they're talking to the cell matrix in all in all directions, and also the cell-cell interactions are in all 3 dimensions. So, and this will affect the shape and the physiology of the cells as compared to 2D model systems where the cells are really in just this flat planar surface. They're only talking to each other in the XY axis, so only on the sides, and they're only talking to the matrix in the cell culture on the substrate below them. So you can see that this will affect the way the cells are organized, the way they perceive gradients of soluble cues, and so in a 3D cell culture, you'll actually establish gradients, whereas in a 2D cell culture, you'll equally bathe the cells in a soluble cue, which may be beneficial if you're interested in a uniform biochemical signaling pathway, but not beneficial if you're interested in the physiological response of the tissue. Moreover, in 3D cultures, you'll get enhanced cell polarity and even tissue polarity with a basal cell surface and apical cell surface as well. And so there are a few different 3D cell culture models, the most popular being spheroids, organoids, and 2D, 3D tissue hybrid systems, which are based on air-liquid interface cultures. So spheroids are probably the simplest system. They're multicellular clusters, typically generated from primary cells or cell lines. A good example of this is the Hep2G cells, which are used to model liver-like spheroids, and they naturally aggregate into these multicellular clusters, and they are free-floating in the liquid media, and they can provide some nice physiological readouts, at least compared to 2D systems. And they're scalable for high-throughput screening, which makes them beneficial for drug testing. But the thing to consider here is that they, since they're derived from cell lines or primary cells, there's some drawbacks as well. If they're for the Hep2Gs, it's a cancer line, so it could give some off-target effects as well. Organoids in the center here are multicellular self-assembling mini organs, and these have really taken off in the last 10 years. They are cultured in a 3-dimensional matrix, such as Culturex BME or collagen. They tend to be generated from tissue, directly from tissue biopsies or from pluripotent stem cells. And when they're cultured, they have a compartment, a stem cell-like compartment, which then generates the tissue and the differentiated cells of that tissue. So the classic example is the intestinal organoid, where they form actually crypt-like structures, which contain the stem cells that then generate the various types of cells in the intestine, goblet cells, enterocytes, and Paneth cells, for example. And so these organoids recapitulate in vivo organ architecture quite well. So if you look at a cross-section of an organoid and compare it to a cross-section of an intestine, for example, you'll see some similar features. And this makes them amenable to personalized medicine and also to high throughput studies, but perhaps not as high throughput as spheroids. Lastly, on the right is an example from ALI culture system, so air-liquid interface. And these have been used for organotypic cell culture preparations for some time. They've also been used in skin biology as well as lung. And an example shown here is is a schematic from skin, and they really recapitulate the in vivo architecture quite well with the cell layering you'd expect to see with a basal cell layer, a stratum granulosum, stratum spinosum, and a stratum corneum. So you really get a nice reflection of the in vivo environment. And the great thing about these cultures is that they are really 3D tissue on a 2D surface, so they're accessible, and they can be used for high throughput analysis as well. And our MimEX system is actually based on this system as well. So organoids have taken off with protocols developed by Hans Clevers, Calvin Kua, Bissell, and others. And there's a lot of protocols out there, which is good because there's a lot of variety, and you can tailor these protocols to your particular needs. At Bio-Techne, we offer many of the components that you need for these cultures, including high-quality bioactive growth factors, as well as small molecules and media components. And so the field is developing, and this is good in that you can establish your own protocols using and tailor your protocols according to your needs. And so you can use different resources, look in the literature, or come to our webpage for a good starting point. We have an Organoid reagent guide, and in this tabulated spreadsheet, you can click on the organoid of interest, such as intestinoid or liver organoid, and it'll direct you to base media components and growth factors and small molecules that you would need for these types of cultures. And this is a good starting point. Another good resource is the Organoid Resource Laboratory from Travagen's website, and this actually provides detailed protocols as well as helps with the media formulation and getting started in the organoid culture realm. So at Bio-Techne, we're also interested in optimizing organoid cultures for ourselves and for other researchers. An example of this is on the left side here. This bar graph is from Travagen, and you can see that the intestinal organoids are normally cultured in Culturex BME type 2. This is quite widespread in the scientific world, and they grow quite well, and the take rate and health is good. But recently, Travagen developed this type R1 Culturex, which has improved levels of intactin, which increases the cross-linking of these extracellular matrix components. And there's a nice increase in the health and take rate of the organoids. So that's something to consider is your matrix and what matrix you're going to use in your organoid research. And on the right, at R&D Systems, we are interested in developing more chemically defined media and improving upon differentiations that are used to make organoids. And in this case, we compared a competitor supplement to our N21 supplement, and you can see here in green that the differentiation of pancreatic organoids is improved using N21 max compared to the competitor, and this is shown by PDX1 expression. So this is another area where we're trying to improve upon organoid cultures. So the benefits of organoid cultures are in its structure. So the 3D structure models in vivo tissue better than 2D models. They are amenable to high throughput analysis, and iPS cells can be used as a starting point. So these induced pluripotent stem cells are great because they provide an isogenic source for many different types of tissues. So if you're interested in studying a disease such as cystic fibrosis that's affecting multiple organ systems, you can derive your tissue from the same patient and study the mechanism of the disease in these different tissues. And so that is a definite benefit. And it's a rapidly evolving field, so you're in good company. There's a lot of literature out there and possibilities to learn and grow. The challenges from the organoid field are the difficulty in culturing. So some organoids are easier, such as mouse intestinal organoids are considered relatively easy to culture as compared to other organoids from, for example, human colon organoids are a little bit more difficult. So this is something to consider. You have to have a robust protocol, and so the take rate, viability, and tissue source could be a limiting factor. When using induced pluripotent stem cells, you also have to have a robust differentiation protocol for your tissue of interest, because you may end up with immature phenotypes or off-target cells if you differentiate your cells using a protocol that is not robust. Another problem with the organoids is the variability. So in a single field of view, you can see organoids with various shapes and sizes. You may have a nice intestinal organoid with crib structures that's relatively large right next to another organoid that's small and spherical. And this could provide difficulty in reproducibility. Another issue is the accessibility of the epithelial surface, because the lumen is enclosed in organoids. So this brings me to the MimEX Tissue Model System, which we've been developing based on work by Hua Zhang and Frank McKeon at the University of Houston, whom we licensed this technology from. And it really builds upon the advancements of organoids, but provides an accessible, easy-to-use tissue system. So it's an alley-based system, and it's a 3D culture system on 2D. And it's for the generation of ex vivo epithelial-derived organ tissue with benefits including in vivo-like architecture and physiology, accessible epithelial surface, consistency in the tissue size and differentiations, as well as having a sustainable source of tissue production in adult ground state stem cells. So this technology is applicable to many types of organs, epithelial-derived organs, including the GI system, which is what we've been focusing on, and as well as liver, lung, and pancreas as well. So it's not only applicable to different tissue types, but for various different applications, including developmental biology, disease modeling, drug validation, biomarker discovery, and toxicology. So how does it work? So in the beginning, you need stem cells. So you need to isolate adult stem cells. You can do this with a biopsy and using a tissue processing kit and really a relatively simple protocol, isolate the stem cells. Or you can start directly with stem cells that we have isolated for you. And you proceed to a two-dimensional culture system where you can expand these stem cells in clones, and they can be propagated and expanded, frozen down. You can perform experiments on the stem cells themselves. You can do gene editing. And from this point, you can then go back to your tissue of interest and differentiate the stem cells into 3D tissue using a specialized differentiating media and an air-liquid culture system. So what does this look like? This is taken from Wang et al. From 2015 from the labs of Frank McKeon and Hua Zhang. And you can see on the left are nice colonies of intestinal stem cells, which are highly proliferative, shown by Ki67, and express stem cell markers like SOX9. And in the center, you'll see a bright field image of an alley culture where you have nice invaginations that resemble intestinal folds. And on the right, you see some IHC showing the cell types that you would expect to see in the intestine, such as goblet cells, Paneth cells, enteroendocrine cells, as well as a marker for apical polarity, villain. And here's a couple examples from our lab where we isolated stem cells from human ascending colon and human descending colon, and we're able to differentiate the tissue and got nice markers such as MUC2 positive goblet cells shown in red on the top. The system is robust and can be used essentially on any region of the GI tract. Here's an example from ileum, and here's a way we like to track our differentiations and look at different markers using a system called PEGI-SU, which is a multiplexed Western blotting system, an automated system. And here in this example, we have 4 lanes, so 4 samples, including the stem cell sample, and then alley differentiation day 5, day 9, and day 12, going from left to right. And you can see that SOX9 expressing stem cells are high in the stems and the 2D culture, but then decrease over time with the differentiation. And there's corresponding increase in expression of Paneth cell markers like lysozyme, SPINK1, and defensin 6A. And there's also increasing expression of enterocytes shown by APN expression. So by day 9, you have good differentiation and even better by D12. Another way to look at this, which is much simpler, is to just use your phase microscope, and you can see that the differentiation progresses with infolding of the tissue and then the formation of complex intestinal folds, so that by day 9 you have nice differentiated tissue, at which point you can perform your experiments. So MimEX offers numerous benefits, and these I will go into detail in the following slides. This includes the accessibility, the consistency of the tissue, the regional specificity, the recreation of diseased tissue, as well as the possibility for gene editing. So MimEX tissue can be considered like an organoid that's unfolded, or an open-faced organoid. So it really makes the luminal surface of the epithelial layer accessible for experiments, which organoids are enclosed in the lumen, so it makes it very difficult. And this, it can be crucial for epithelial tissue, especially intestine, where the barrier function is perturbed in diseases such as infections or in irritable bowel syndromes like Crohn's disease. So being able to assess the function of the barrier can be critical for studying these diseases. So here's an example of a 96-well formatted air-liquid interface culture. So this is amenable to high throughput as well. You can perform tear measurements, but in this case we used fluorescently conjugated dextran, which you add to the top of the well of your transwell, and then you can wait for it to diffuse through to the bottom layer, which you then will take and read for the fluorescence. And you can see that with transverse colon tissue, there is a very nice tight barrier, in which over 24 hours about 1% of the dye passes through. And you can then study what affects this tight barrier. And in this case, as proof of principle, we used a strong actin depolymerizing drug, latrunculin A, to disrupt the tight junctions and cell adhesions, and this increased the permeability. And so in this 96-well format, you can screen different conditions and look for the effects on barrier function. Another benefit is the consistency of the tissue. So our tissue arises from a monolayer of stem cells, and then from there, there's a stereotypical differentiation that occurs, and this is consistent from well to well, and with the size and shape of the well determining the size and shape of your tissue. So this is great for reproducibility, and this may be a drawback for other 3D model systems like organoids. Another important benefit is the regional specificity. So the stem cells retain the memory of the site from which they were taken. So for example, on the right, if you are looking at the airway system, if you derive stem cells from the distal airway, as shown here in red, you will then differentiate into type 1, type 2 alveolar cells and Clara cells. On the other hand, if you isolate the stem cells from the proximal airway, shown on the right with this bluish purple color, you will derive the tracheal epithelium, so a high number of goblet cells and ciliated cells. And this is really great if you're looking at regional specificity of certain drugs or toxic compounds. And this is shown in the Kumar paper from 2015. Another example of this is taken from the Wang paper in 2015, where they looked at different regions of the intestine. So they isolated stem cells from the same individual from the duodenum, jejunum, and ileum. And if you look at the gene expression profile on the right, you can see that there's very specific gene expression signatures for these regions. They have overlapping gene expression as well, which is general for intestinal function, but these specific changes or differences in gene expression could reflect the differences, the subtle differences in the function of these different regions. And you can see this regional specificity when you do the air-liquid interface cultures. So the duodenum tends to form this thinner, less complex types of tissue, where the ileum is thicker with more goblet cells, and this is apparent in these differentiations. And if you do colon, there are even more goblet cells, for example. So they really retain their characteristics of the site of origin. And another benefit of this memory of the stem cells is that you can recapitulate disease. So if you have a genetic or epigenetic mechanism underlying a disease, or a disease which changes the stem cells, you can recapitulate this in ex vivo using our system. So for example, in Crohn's disease and in other cases. So this makes it ideal for disease modeling or biomarker discovery, drug feasibility, and toxicology. So the idea is that they maintain this memory of the tissue of origin. And a good example of this is taken from this Yamamoto paper from 2016, looking at Barrett's esophagus. So Barrett's esophagus is a intermediate phase for esophageal adenocarcinoma, so esophageal cancer. And in order to study the mechanisms of this disease, they isolated the stem cells from the esophagus, from the Barrett's inflamed region, or the Barrett's lesion it's called, and from the gastric region. And they grew the stem cells and performed these differentiations on certain clones from the Barrett's esophagus. And on the bottom, you can see the differentiations with H&E staining. And on the left, you see esophageal tissue is normally this stratified squamous epithelium, which is stereotypical. And on the right, you see the gastric, which forms a little bit different columnar type epithelium. And the Barrett's esophagus in the center is actually a hybrid of esophageal, gastric, and intestinal tissue. It shows features with these invaginations, and it's highly hyperplasic and abnormal. And they performed gene expression analysis and saw that they had upregulation of genes that were in fact intestinal genes. And this was quite interesting. And they then compared the Barrett's esophagus gene expression profile to more advanced esophageal cancer, and they saw some of the same changes. So on a principal component analysis, they would cluster the same, except for the expression, the changes in oncogenes in P53, which were absent in the Barrett's esophagus. So it really suggests that Barrett's esophagus is an intermediate or a precursor to esophageal cancer. It also suggested why it's difficult to get rid of Barrett's esophagus, which sometimes they'll remove the lesion and it'll regrow. And this work suggests that in order to stop this, you have to target the stem cells and get rid of those as well. So again, we have the maintenance of disease, and we did this in our own lab as well. In this case, we got a biopsy from a lung cancer patient in which we derived the stem cells from the cancer tissue as well from normal adjacent tissue. And on the left, you'll see the allele differentiation from the normal lung. You have this nice KRT5 positive pseudo-stratified columnar cells that are low in P63. On the right, you see this stratified squamous epithelium, which is high in P63, a cancer marker. And this is typical of many types of cancer. And this reflected what we saw in vivo. So from the same patients, we had tissue blocks. And when you look at the P63 staining, you see this increase in P63 with this stratified squamous epithelium in the cancer, but not in the alveola of normal lung on the left side. The final benefit of the MimEX tissue system is the ability to clonally expand and gene edit the cells. So you can take a single cell and grow a clone, which then can be used to expand pedigree lines. And with this single cell, you can perform CRISPR-Cas9, you can correct a genetic mutation, for example, and derive cells all with this corrected genetic mutation. And this has implications not only in studying disease, but also in regenerative medicine as well. So here is an example from the Wang paper from 2015. And this just shows that you could take a single cell, grow nice colonies, which can be further propagated. And on the right in 96-well format, you can see that the success rate ranges from just below 50% up to above 80%. So the ability to grow clones and make pedigree lines is quite robust. So currently we have the GI system out, MimEX GI, including all the media that you would need, the expansion media, differentiation media. We also provide cell lines. Currently we're only offering the descending colon, but we're working on other lines which are forthcoming. And we offer a starter kit and tissue collection kit as well. And it's not only the GI system we're interested in. We are interested in other epithelial tissues. Currently, we are actively working on airway, liver, and skin, and these will hopefully be out within the next 6 months to a year. So in the end, just to bring it back to a little bit of what Scott was talking about, you need a culture system to answer the particular biological question you're asking. The solution may be a 2D culture system using induced pluripotent stem cells, or it may require a more 3D-like tissue system, which are more predictive for drug efficacy and toxicology. So the system that you need needs to match your particular biological question, and Bio-Techne offers solutions for all of these particular models. And MimEX is a good compromise between complexity and predictive power, as well as ease of use. So with that, I am finished, and I thank everyone for tuning in, and I believe we have time for questions after this. So that's all I have. Thanks.
Thank you, Dr. Flynn. The audience has submitted several questions, so let's get right to them. Dr. Flynn, this first question is for you.
Okay.
Where in the spectrum of model systems would you place organ chips like those made at Harvard's Weiss Institute? How would it compare to MimEX?
So the chips are very nice. So the organs on chips are complex in vivo-like models. So the ones from the Weiss Institute and Emulate are very nice. They've been shown to be predictive, but they tend to be pretty expensive and more difficult to use. You need a flow system, and so I've seen some data from it, and it's great. So they appear to have a very strong predictive power, and so somewhere on the spectrum here, I think at the end I mentioned it. They're a great model system. So we believe that MimEX can be comparable to tissues on chips. The one thing that MimEX doesn't currently do is multiplex multiple organs on the same chip, but we're working on this, and this should be possible in the future as well.
Great. Thank you. This next question I believe you could answer as well, Dr. Flynn. Is the size of a 3D system a limiting factor in the study of human development? What is currently the largest size of a MimEX model possible?
Oh, that's a good question. So currently, so does size matter? And there is some suggestion that size does matter, and that modeling human development in human tissues, there is a size component. And one thing that is extremely important is, at least with multiplexing these organs, is the ratio of the size of the organs. So in terms of the smallest size, I don't have an easy answer for that. So for MimEX, we use 96-well format, which are, I don't know the exact diameter here. I mean, we're talking a growth area of, very small. I mean, so our 24-well is about 1.5 centimeters square, something in that line, 1 to 1.5 centimeters square. You can also use 12-well or 6-well alleys. So we haven't gone above that. And whether or not this accurately models human tissue, I think we get a lot of the physiological function even on a 96-well format, especially in terms of like the barrier function, the mucus production, some of the immune response that you might see in the tissue. So how small can you go is a good question, and I think MimEX it's applicable to various sizes, and it depends on exactly the experiment you're looking at to perform. But even in 96-well, we do see some function retained. So I hope that partially at least answers the question.
Yeah, Kevin, just if I can add into the MimEX system relies on an air-liquid interface, so the transwell systems. So really the limiting factor for using MimEX in the size, tissue size creation is the smallest size you can get an effective transwell, which I think at this point is the 96-well plate.
Thank you both. Dr. Schachtel, this is actually sort of a follow-on question from that. Can you explain how one cultures in the air-liquid interface? Is there a particular type of plate that's used with MimEX?
Yeah, sure. So so the air-liquid interface involves using a transwell insert, and currently we use Corning transwell insert plates. And so this is simply like a 24-well plate that has like an insert cup that sits inside each well. And the transwell insert has a membrane at the bottom that allows media exchange from the underside of the transwell, and then leaving the inside or the upper surface of the membrane exposed to the air. And so for the MimEX tissue, what we're doing is we're taking the adult stem cells that have been expanded, plating them in a high-density monolayer on top of the membrane on the insert. And so it's getting fed culture media from the bottom and then exposed to the air on the top. And that orientation of the air-liquid interface helps define the direction for the differentiation to define the apical to basal compartments. So I hope that explains it a little bit more.
Great, thank you. Dr. Schachtel, this question is for you as well. Are your live stem cell imaging kits shown or demonstrated not to impact cell behavior? You are, after all, interacting with the cells via antibody.
Our research and development team has gone through lengths to make sure that this is not the case. So they've gone through experiments where they've placed the glow live antibodies and visualized the staining on the cells, and then measured their ability to continue to proliferate over time compared to an unstained population, showing that there's no difference in proliferative ability. And similarly, taking those stained cells and differentiating them down the line, they've also shown that isn't impacting compared to unstained cells the ability for them to differentiate. And I believe there's some data supporting that on our website on the product page.
Thank you, Dr. Schachtel. This question is for you. I'm not sure if you can answer it directly. The question is, does R&D Systems offer a marker for mature cardiac pacemaker cells? So I would extend that to say, how would someone go about finding that info, in case you don't have it off the top of your head?
Yeah, so what R&D Systems is also known for, in addition to some really high quality reagents, is the information that we provide the research community on our website. So there are a number of different ways you can go in and try to find, if you're unfamiliar with what sort of cardiac pacemaker, like a marker that would identify cardiac pacemakers, there's resources on our website defined by research area that you can go through and dig into, and it'll list cardiac-specific markers, and then you can look in there and try to identify a pacemaker marker. In addition, we do have these interactive pathways on our website, which people find really useful. And in the case of this one, for cardiac markers, probably the easiest way would be to look at our induced pluripotent stem cell pathway, which then shows like how growth factors and small molecules and components that would influence the differentiation of these stem cells into different terminally differentiated cell types. And I know one of them is the cardiomyocyte cell. And so clicking on that cell will then pull up all sorts of information in terms of growth factors and markers and such that could be useful for identifying antibodies for, let's say, cardiac pacemakers.
Thanks for that. Dr. Flynn, for organs that have liquids in their lumens, such as kidney tubules that have renal filtrate, is it possible to have another liquid in the top portion of the transwell?
Yes, absolutely. So this is, yes, is the bottom question. You can leave the liquid in the top part of the transwell, and what is the benefit of this over a normal 2D system is that you still have the transwell, which is which then can be removed in section for histochemistry. Also, so this is something I'm actually working on currently with the liver system, which also is not actively exposed to air either. And whether or not this is beneficial with having liquid on the top versus air is something that we're currently studying. And so it definitely is possible. In the case of intestine, it does make a difference. The differentiation does improve upon the removal of the liquid on top. So in airway, for sure, it's much better to have the air-liquid interface rather than a liquid-liquid interface. But it's certainly possible. We haven't done any tests with kidney yet, but we look forward to looking into that as well.
Thank you. Dr. Flynn, this one is for you. Are organoids or MimEX grown models implantable? This question is coming from someone who's interested in the effect of the tumor microenvironment on brain organoids.
Are they implantable? So can we take them and then put them into a tissue or into a nude mouse, for example? And the answer is yes, it's been done before. So people have done this, and in MimEX Tissue Models, our collaborators have done this and shown that you can get the differentiation in nude mice as well. So in terms of tumor models and brain, or specifically for brain, we have not performed these, but it's worth looking into if these in vitro grown tissues, these cancer generated tissues are implantable and which exhibit metastasis in vivo. We haven't done that yet, though.
Thank you. Dr. Flynn, this is another question for you. Is it possible to isolate cells via liquid biopsy or circulating tumor cells as the starting point for a MimEX culture?
Oh, that's a great question. Theoretically, it's possible. We have not tried. So that is a good question, and we have managed, or at least our collaborators at University of Houston have managed from very small biopsies, from endoscopic biopsies, so very, very small pieces of tissue, but from circulating cells or circulating tumor cells, for example, I'm not aware that we've tried. We've only tried from solid tumors and from solid tissue samples, but it could be theoretically possible, but we just haven't been able to try.
Thank you. Dr. Schachtel, this question is for you. How reproducible are assays using patient-derived iPSCs? What can be done to help standardize?
Yeah, it really, like I mentioned throughout my talk, really vetting your iPSCs at the start of your experiment before differentiation can be an important component to creating good reproducible models. So making sure they're expressing pluripotent markers and even more importantly, making sure that they can differentiate into the cell type efficiently that you're looking for. Using the functional identification kit can allow you to look at different iPS cell lines and look at which ones are more prone to be able to better differentiate into that tissue. So really looking at the front end and validating your iPSCs on the front can help out. In terms of, I think there's always going to be, if you're looking at patient-derived iPS cells, there's always going to be variability between them. And so there's going to be, I think the field can evolve still in developing more standardized protocols to do this across the different labs as well as across different stem cells. So getting more robust protocols, and that's some of the goals of what we have here, what we're trying to do here at Biotechne with our StemXVivo kits is, we're testing those on a number of different patient or human-derived iPS cell lines to make sure that like the kit isn't just working on the particular one that we started testing it on, but that it works across multiple cell lines, and trying to optimize the system so that will do so. And I think that if you're doing this in your lab, that's going to be an important point for you to look at as you do it as well.
Thank you. Dr. Schachtel, this question is for you as well. How long are iPSCs culturable in your models?
Actually, this might be better for Kevin to answer. I mean, iPSCs that we use, I think we can culture them for an extended amount of time.
Yeah.
Go ahead.
So I'll just jump in real quick. So yeah, we culture iPSCs for, I mean, I haven't gotten to a limit, but this is a question of how stable they are and how stable is the genome. And we typically, after about 2 to 3 months, we tend to stop just out of concerns that with passaging and time and culture they may accumulate mutations. But it has been shown that they can be, with the right conditions, quite stable. So how long is a good question. They will accumulate mutations, and this could affect their physiology later. So months and many passages, they are genomically stable. We've actually done a little bit of studies of this, but we haven't gone beyond a few months. So it's hard to say exactly when, what's the limit.
Okay, thank you. And now we have time for just one more question. Dr. Flynn, this one is for you. Is there a brain cell-derived MimEX model in the offing? It may be very useful for the study of autism or epilepsy.
Okay, so there isn't yet, but we do believe that it is applicable. So it's not on our immediate radar, but we believe that we will be able to isolate brain stem cells as well using the system. So it's something our collaborators have at least done on one occasion, and so it is possible. And we may work towards it in the future, but currently there's nothing available.
Well, thank you. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses will be shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Scott and Kevin, as well as our webinar sponsor, Bio-Techne. Thanks, everyone. Goodbye.